Okay, thank you first of all very much for the kind invitation to speak to you this morning. It's a very daunting task to give a keynote lecture in any symposium, and especially in one like this, which is an interdisciplinary symposium where the vast majority of you are coming from other disciplines than biology. When one gives a talk to the general public, if they don't follow what you say, then they just think that they're stupid and that they should go away and read and learn more. Whereas when one talks to fellow academics, if you don't understand what I say, then you're perfectly well aware that it's because I didn't explain it properly. And so I'm going to try to keep things as simple as I can, but also to try to speak as far as I can, or at least attempt to speak your language so that we can have a meaningful dialogue, which is the whole purpose of the event. Most of the world's religions focus on the idea of mankind as the pinnacle of creation. And our mission here on the planet, either to be custodians of the rest of the created universe, or to improve it, perfect it, and make it in some way more like the divine that inspired the beginning of everything. However, as a biologist, I'm coming to this in a different way, because our collective view is that humans, even though we are very important products of evolution, we're not the reason why evolution happened, and we're not the only outcome of evolution, and we may not, and almost certainly will not be here forever. So there may be other life forms that take our place in the future, just as we have taken the place as the dominant species on the planet from the dinosaurs that were around 100 million years ago, or the bacteria, of which I'm going to say quite a lot in my talk, which have been here pretty much all the time. I'm going to depart from the traditional format of a keynote lecture, because I think nobody wants to hear somebody drone on for an hour and a half on things that they don't understand. And so to promote dialogue, what I'm going to do instead is split my talk into 6, not necessarily even, but subsections of the topic. And at the end of each, I'm going to pause and ask you to reflect on what I've just talked about and ask questions that come to your mind. And if they don't come to your mind, I hope that I will be able to provoke a few questions. On the other hand, you might, well, hopefully will ask some questions that are coming up, or the answers to which are coming up in the following section. So these are my subtitles of the 6 sections, which I'm going to go through. And they break into 2 halves, really. The first half is the main part of my talk, where I'm going to talk about the history of life on this planet. If the theme of the symposium is becoming, these are going to be about what we are and how we became what we are. And then the final 3 sections, I'm going to reflect on the future dimension, what we are becoming and what we will become in the future. But the we that I impose here is not necessarily only referring to humans. So I'll start with the history of our planet. I could go back further and talk about the history of the universe, but I'm not a cosmologist, so I don't think I'm qualified to do that. When we talk about the history of the planet, we have to take account of many different disciplines, or studies in many different disciplines: cosmology, planetary sciences, geology, the sciences of the atmosphere, and then we come to biochemistry and genetics. And I said I'm not an expert in any of these things until we get down the list. So anything I say in regard to cosmology and the origins of the universe may be out by 1,000 million times in terms of scale, whether of time or of space, and I hope you'll take what I say to be at least poetic rather than literal. Okay. So let's start with just considerations of where we are in the universe. What is the scale of the universe? So there are things that we're familiar with in an everyday sense that we can relate to, the environment in which we live, the size of the things in our life, the people, the animals, the buildings, the plants, and so on. And we can just about grasp the size of the solar system, which is 3 times 10 to the 9th kilometers in diameter. That's a very arbitrary figure because there's a lot of debate, at least as far as I understand it, in the field of planetary sciences as to what should be considered the boundary of the solar system. 3 times 10 to the 9th is 3,000 million kilometers. And that's the distance out to the farthest dwarf planet that's been identified so far. But that's only a tiny fraction of the distance traveled by light in a year. The galaxy in which we live, the Milky Way, is 100,000 light years in diameter. And when we consider the size of the known universe, we're at many orders of magnitude greater than that. And so our ability to know what's out there is, of course, very limited. And similarly, in terms of the time scale of the whole universe, if we consider the Big Bang as the origin of the universe, at least the origin that we can relate our current universe to, that's about 14 billion years before present. Our solar system then is relatively young in cosmological terms, and the first life appeared only 3.5 billion years ago in the form of the first cells. Before the first cells, there were chemistries around on the ancient Earth that did some of the things that we regard as life, but the whole package came together as in the form of cells 3.5 billion years ago. But it's only 650 million years ago that we see evidence in rocks of the first animals, and the first modern humans only 150,000 years ago. And then our own experience is much shorter than that. So things that we know that have happened in real historical time are a tiny fraction of that human history. So how can we study all of this? Of course, we can't go back in time. We can only look at what is there now to figure out what used to be. When we actually look at distant stars and the edges of the universe, we are actually looking both across the distance of space and time, because it's taken light millions of years to reach us from those farther extremities of the galaxy. So we're actually looking at the universe, or the outer reaches of the known universe, as they were a long time ago. In terms of what we can extrapolate from the known physical universe we have here today on this planet, we need to look at signatures indicating the processes that happen in history, and largely that means we have to look in the rocks to look for the relics of living organisms, fossils. However, we've realized in the last 50 years that we can do better than that, because living things not only leave an imprint of how they were when they were alive, but they also, because we are all reproducing and donating our genetic information to the next generation of cells and organisms, we also have an informational imprint in our cells, in ourselves, which tells us about our history. And we can compare those signatures and use them, as I will repeatedly in my talk, to reconstruct the history of what happened and how we are related to other organisms that are alive today and other organisms that were alive a long time ago that are the ancestors of ourselves and all the other creatures and organisms alive today. So one of the most striking things to a biologist about this history is that we're pretty much all made of the same stuff. When I say all, I mean all humans, animals, plants, microorganisms like bacteria, and many other single-celled organisms that I'm going to talk about today. We're all made of the same stuff because we are all made of cells, and the cells of humans and all those other organisms on this planet are basically composed of the same chemistry. The same chemistry directs our ability to derive energy from the environment and our ability to reproduce. It's all based on the same stuff. And so that teaches us that either life originated on this planet or arrived on this planet, we can't say which because we no evidence at this point of life anywhere else at all, but that happened only once because everything stems from this tree of life wherein we are all composed of the same stuff as our ancestral cells 3.5 billion years ago. An alternative idea, which I think we can't disprove, is that there were many other kinds of life around 3.5 billion years ago, and maybe a bit later, but not very much later, but they're all extinct. And the only one that survived is the one built of cells like we are composed of ourselves. We can define life in a more prosaic manner as these three elements, which could apply to many different chemistries that are not the chemistries of living organisms today. We can hypothesize that other chemistries in other planets, even other planets in the solar system or their moons, could have given rise to life which fulfills these basic principles: the ability to replicate, the ability to organize, and the ability to derive energy from the environment to sustain those processes. But I'm going to focus on what we know. We don't know about any other kinds of life than the cells we are composed of, and so I'm going to focus on what we know about that life. The commonality of life as we know it leads to the concept of a single and universal common ancestor of all life on Earth. There's some debate as to whether that really was a single organism as we would recognize it today, or a community of organisms all cooperating and doing different things. That debate is not resolved, but I think the idea of Luca, the last universal common ancestor, is valid whichever model we subscribe to, whether it was a single cell or a community. And it has very specific properties that I'll now enumerate without going to any of the chemical detail. So all cells are basically an aqueous entity. That means that they are built on water. Everything's in solution, just as in this cup. But around that aqueous space, demarcating the space from the outside, is an oily layer which is actually composed of a double layer of molecules that are hydrophobic on the inside. That means that they are oily on the inside, and they are hydrophilic. They are interacting with water on the other side. And that represents, so that structure is called a membrane, and it's found around all cells. Some cells have an extra layer around them for protection or insulation or defense, but that's really a bit of decoration on the basic principle. Membranes define all cells, and all of the membranes defining our cells are basically organized in this same double lipid bilayer structure. Our information, which directs what we are and how we develop and how we live, our entire biochemistry, is all encoded in a universal chemical language in the form of DNA and its relative RNA. RNA is just a working copy of DNA. DNA is written, it's an informational language written in a 4-letter chemical alphabet, and that chemical alphabet is universal. All organisms on this planet use that alphabet and no other, and the specific chemistry of DNA is the same in bacteria and in humans. And even more specific, all cells contain an assembly of components that we call a ribosome, which is illustrated here. It looks like this little blob at the bottom right of my slide. The ribosome is a molecular factory for interpreting the information in DNA and RNA and turning it into the action molecules of our cells that perform all the chemical reactions and form all the other structures than the membrane, and these are proteins. So ribosomes are protein synthetic factories, and all cells contain ribosomes, and no cells make proteins in any other way than using this ancient conserved machinery. Although the bacterial ribosomes and human ribosomes are a little bit different in details, in essence, the chemistry that they do and the way they do it is the same. And then there are a whole set of biogenesis platforms for making membranes, making DNA, and making ribosomes themselves that are also universal to all living things. The genetic code, which enables the translation of DNA information into a meaningful sequence of components to make a protein, that is also pretty much universal. I'll mention a couple of exceptions later on, but all organisms, right from bacteria to ourselves, use the same way of encoding genetic information. The pathways and players in metabolism, the chemistry by which we degrade sugars or build up more complex molecules, that is all the same in all organisms. And the actual proteins, the action molecules that do all that, are similar in all organisms. So similar that we can align the information in this linear way as depicted on the slide. I've made it so small that you can't read any of the text because I don't want you to read the text. All I want you to see is that I've just cut, well, the person who made that particular diagram, because I stole it from the internet, that diagram just shows in a kind of simple color coding the aligned sequence of a particular protein from different organisms. And you can see there are many points where the same color occurs, and there are other points where there's a variant color in one or the other. And we can use this kind of approach to trace the informational relatedness of different organisms. But the essential point that I want to emphasize here is that they're all similar. So a particular process is not only done using the same chemistry, but it's done or it's facilitated by a protein that actually is ancestrally related to all the other proteins that do that job in all other organisms. And then one other universal feature, not only having a membrane, but having within that membrane embedded some little molecular machines, usually built on proteins, that are able to communicate with the outside world. If life were just an aqueous drop inside an oily skin, we would be isolated from each other and we wouldn't be able to interact with our environment either. What the membrane transport functions that we all share do is import materials from the outside that we can eat, export materials from the outside that we can use to signal to other organisms or other cells what we are and what we need. And those processes occur not only in single-celled organisms like bacteria, they also happen inside our own bodies because the different cells in our own bodies need to communicate with each other to inform the neighboring cells what they need. So the liver has to interact with the heart, the brain has to interact with the skin, and so on. I'm going to pause now and ask you to come forward if you have some questions on what I've just presented. And then we'll go on and talk about the first cells and how we got from the first cells to where we are now. Yeah.
Could the speed of evolution possibly be higher than it has been?
As I've already sort of implied, because life as we know it only arose once or arrived once on this planet, we are lacking one of the cardinal principles by which we could answer that question. Because to study anything, whether we're studying cells or music or philosophy, we have to have something to compare it with. And we've only got this one example. What we can say is if we look at different periods during that evolutionary history, it does appear that certain traits developed or evolved at different rates. But we should remember that bacteria, and I'll come to this in a moment, bacteria are pretty much now like they always were. It's only a small group of organisms, including us, that during evolution have apparently changed significantly and become, at least in our case, a little bit more complex. So I don't think we can really answer that, but one of the assumptions when we look at aligned DNA and protein sequences is indeed that they're evolving at a roughly constant rate. And although we've got some evidence of that by comparing we can at least compare the data that we get from DNA sequences with the data that we can get from the fossil record, and by and large they correspond. But on a micro scale, I think, yes, evolution can proceed at different rates, because evolution is, of course, it's not an inherent process to the organism, it's about the interaction of the organism with the environment. And the environment may change very rapidly, or may be constant for millions of years, and therefore the way in which the environment is talking to biological entities may differ in terms of its visible impact.
Okay, thank you.
Okay.
When Nina had his introductory welcome talk, she very much emphasized that when we talk about becoming, we usually refer to what's novel, unique, and what you are doing is kind of the opposite. You are emphasizing the similarities. So why is that so important for you?
I think it's important. Well, it's important for me because that's how it appears to be. But this is, of course, the prelude to the rest of my talk, where I'm going to talk about all the diversity of life and how it comes from that unity. So it's a little bit of a trick that I've just done that to start with, and you'll see in a minute where we're going. Yeah.
I may be a victim of fake news, but aren't there organisms that have been found in deep sea volcanic rifts that actually have a completely different metabolism?
I'm going to talk about those organisms shortly. Not really. I mean, they have adapted to living in extreme environments, but their basic chemistry is very similar. We're talking about small details. I'm going to repeatedly, for some reason, come back to talking about organisms as cars, both in terms of what they look like and how they drive forward. I would consider those to be a little bit like, let's say you could put a track onto a motorcycle and turn it into a snowmobile, but the basic machine is still the same. Yeah.
How about
Yes.
viruses and bacteria? Do they have the same membrane around them?
Viruses are different, and I think there's a lot of debate philosophically about whether we consider viruses to be living. They're not considered as living organisms, but they're part of life. I would regard viruses most usefully as packets of information that cells share with each other, and sometimes they acquire properties that are more destructive of cells than useful to cells. Bacteria are indeed part of the same set of cellular life forms that I've just summarized. So what viruses are? They're small packets of DNA or RNA that code for just a few functions connected with their own survival, but they don't have an independent existence. They have to go to other cells. They actually have to enter other cells to propagate themselves, because all the other functions of cells are there in the cell that they inhabit. They're not inherent to the virus. Okay, I'm going to talk about the evolution of cells. This is just an image of some fossils that are interpreted by geologists to be remnants from ancient cells dating back 3.5 billion years. So if we take that as a likely starting point, and through everything that I've said, that all life forms are united in their overall structure and chemistry, we come to this concept of the last universal common ancestor, which lived 3.5 billion years ago. And if I'd been giving this talk about 45 or more years ago, I would have, based on observation of what organisms are like, I would have divided all the organisms around on the planet into organisms that have cells of one or other of these two types. And in essence, the ones on the left that we call prokaryotes are small organisms, including bacteria, whereas the ones on the right, eukaryotes, are much larger. Bacteria are typically only 1 micrometer in diameter, whereas the cells of eukaryotes are typically 10 to 100 micrometers in diameter, so they're much bigger. And in addition, the eukaryotes, their name means that they have all of their genetic information bundled together in a specific place in the cell called the nucleus, that I've colored here in dark blue, which distinguishes them from prokaryotes, which just carry their genetic information inside the general gazpacho of the bacterial cell. However, from the late 1970s onwards, driven in particular by one evolutionary biologist in the US, Carl Wurzer, we've come to a different view. We now recognize that at least what we previously called prokaryotes, which are these small organisms without a true nucleus, are actually representatives of two different derived sets of organisms that are both related to the universal common ancestor, but they share genetic information and chemistry about specific details of how their membranes are built that distinguish them one from the other. And we've given two names to these. You can quibble about what these names are and whether they're appropriate, but they're now pretty much universally called bacteria and archaea. And I've just picked these fanciful images from the internet to make the point that even though they are ancestrally diverged, they diverge from each other, we think, pretty close to the time of the last universal common ancestor, but they seem to be rather similar. They look similar under the microscope. They do similar things. I'll talk about their lifestyles in a little bit more detail later. So we can say that all life forms on the planet, even though they seem superficially rather similar, they fall into one of these two tribes, which can be considered two domains of life or two branches of the tree. They're prokaryotes in the sense that if we look at all the old Fords, we look at those and we can say, well, that's a car, but it's a Ford because it's got certain features that we can recognize as a Ford. It still comes from the same basic principles of building a car in the same way as prokaryotes, bacteria, and archaea share those common cellular principles, but nevertheless it's got certain extra little features which make it a Ford. On the other hand, if we see another car that looks like this, we know that it's a Volkswagen. And Volkswagens and Fords have been separate throughout their existence, even though they come from a common set of design principles. So in the same way, bacteria and archaea have been separate for 3.5 billion years. Where do they live? Bacteria, in the main, occupy what one might call a temperate environment. They live in the green and pleasant land around us. They're found in the soil, they're found in the ocean, they're found in lakes. They're also, very interestingly, found in our own bodies. In fact, you might be shocked to know that most of the DNA in the human body is not human DNA at all. It's DNA from the bacteria that are living principally within our own digestive system, and which are almost certainly contributing a lot to our own biology. But you could consider that as another temperate, reasonable environment where you might find bacteria. On the other hand, archaea seem to have been squeezed out from these temperate environments, or perhaps they never ever inhabited them because they might have been highly adapted during early evolution to live in extreme environments that are much more like those that pertained on the Earth during its much earlier history. This is an aerial photograph. It's not mine. I've got some crappier ones that I took. I was able last month to take a week's vacation in the US, and I went to Yellowstone National Park, and this is one of the most famous landmarks in Yellowstone, which is the Grand Prismatic Spring. But all this color that you can see from the air, and you can actually see it from ground level, is made out of the organisms that live there in communities, which are almost all archaea. Archaea, in this case, are living at high temperature, but there are other groups of archaea that live at high salt, or low oxygen, or high pressure, or live in all kinds of environments that we would find very inhospitable. Very importantly, although there's been a few minor exchanges of genetic information between those two broad groups of prokaryotes, those exchanges of information are exceptions to the rule. Pretty much all bacteria contain genes and chemistries that are bacterial, and all archaea contain archaeal genes and chemistries, and so they don't mix. They just coexist and to a large extent inhabit different environments on the Earth. What about eukaryotes? We are eukaryotes, and they are something else entirely, a genetic and cellular mosaic. And this has given rise to a lot of debate and a lot of uncertainty about the early steps in evolution that led to us. So this is a revised version of cellular evolution that I think is closer to the truth, but still contains some uncertainties, and so I've put some question marks and some dotted lines here. Eukaryotes, which are indeed all the big cells, of the organisms composed of big cells, they are, based on their genetic information, a kind of hybrid. They contain segments of DNA, genes, and proteins that are encoded by those genes that clearly relate to some from archaea, but also some that clearly relate to bacteria. The most generally accepted view is that we are probably, or eukaryotes are probably, an early branch from the archaea, because we are a little bit more similar in many features to archaea than we are to bacteria. But at a certain point in the evolution of that group, probably when they were just blobby cells that weren't very different from archaea that were around at the time, they acquired some new properties. Some of those properties may be the ones that define them as eukaryotes, but others are ones that they acquired by forming a coalition with not just one, but several different bacterial species that were alive at the time, which had evolved very efficient chemistries to do things that were useful to eukaryotes. So you can look at the eukaryote as a kind of best of both worlds evolutionary solution. Evolution had proceeded independently in bacteria and archaea, leading to some specialized chemistries, and eukaryotes then gobbled up those different organisms and combined them into one to be more versatile, because it then could do lots of those different things and be, in the end, more successful in some ways, although it's debatable whether we eukaryotes have been more successful than the bacteria and archaea that are still around today. So this leads to the idea that there are really three domains of life, and one of the debates here is whether the archaeal part of our genome and our cells is in fact ancestral or acquired from several different groups of archaea, and there is one school of thought which holds that there is another third group of genes that are not very many that are really eukaryotic and are really a third branch of life from the very beginning, and then that group of organisms has just gobbled up these others. I think that's unresolved for now, but the main point is that we are a mosaic, and we have to interpret everything that came after that in eukaryote evolution in terms of that mosaic and what that mosaic is composed of. All right, so before I go on, I'm just going to survey very quickly what makes a eukaryotic cell a eukaryotic cell. I've already mentioned its greater size. Because it's much bigger, so it's 10 or 100 times bigger than the largest bacteria, and that means it's got a volume that is from tens of thousands up to millions of times greater than that of bacteria. It's got a lot of internal structure. It's got a filamentous cytoskeleton, an architecture of the cell inside it made of proteinaceous filaments that give it shape and to a certain extent rigidity and enable it also to move. It contains organelles, particles within the cell, and I'll come to the details in a moment, which carry out specific biochemical tasks. It has an endomembrane system, so it's not just got a membrane on the outside, but it's got membranous structures right through the cell which are interacting with each other and with other organelles. And one of those organelles, which is connected to this internal membrane system, is the nucleus, which contains the genetic information, which is now separated from the rest of the cell. A very important property of eukaryotes, which is shown in a primitive form by some bacteria, but they may have been the group of bacteria that were taken up by eukaryotes to create this mechanism in the ancient eukaryotic cell, and that is the process of endocytosis. So instead of just having proteins in our membranes which can absorb small molecules from the outside and export molecules to the outside, eukaryotic cells have developed a property whereby they can engulf with their membranes a huge blob of the outside world, take it inside and then process it, eat it, or interpret what molecules are contained in that blob of stuff that it's sucked in from the outside to try to figure out how it should respond to its environment. Is there something nasty out there that needs to be defended against, and so on. The actual genome, the DNA of eukaryotes, is very different in terms of its organization and its size than that of bacteria and archaea. I would draw this analogy. Bacterial and archaeal genomes are very well organized, like the perfect modern office, whereas eukaryote genomes are much more like a typical academic's office, looking like this with piles of papers. Nevertheless, most of you will say, if you have an office like that, and I guess most of you do, you will nevertheless assert to your students and to each other and to me that you can find the crucial document within that mess of paper anytime you want, and that really there's a kind of organization in that chaos. But that's how eukaryotic DNA is organized. Our genes are scattered around in pieces all over the place. The genes that perform a common function might be even on different chromosomes, which are structures that contain genetic information. And so the whole thing looks superficially a mess, but somehow that's organized in some way. And then another property that's unique to eukaryotes, but it's not shared by all eukaryotes, is the ability to be multicellular. That's very important. We're composed of 10 to the 11th, 10 to the 12th billions of individual cells that are all in some way interacting with each other to make our bodies. That's true also of a plant or a tree, for example. But there are also some eukaryotes that are single large cells which have many properties of other single-celled organisms that are not eukaryotes, but multicellularity evolved only in the eukaryotes. And then sex, which I just want to define very quickly, is something that there's some primitive echoes of sex in the prokaryotic world, but sex as we know it is pretty much universal in eukaryotes, and I've depicted it here without any complex terms, so most people who talk about sex will just throw lots of Latin and Greek words at you and get you very confused. The basic principle is that during the course of evolution, a species, group of organisms, in this case I've simplified it down to a single-celled organism with just one packet of genetic information, one chromosome, will develop mutations, changes in the genetic information that make it similar to all the other cells of the species, but still unique. And I've therefore depicted those two different forms as green and red. And sex involves those two cells with slightly different information fusing together, bringing their information together, and then using both sets of information to do all the things that that organism is going to do. So it's got two bites of the cherry. It's got two alternative ways of doing anything that are rather similar, but they are nevertheless genetically non-identical. And then all types of sex involve exchange of information between those two genomes to create hybrid variants, so new sets of genetic information that contain some information from each of the original parents, is then formed by a process known as recombination. And then that cell undergoes a series of divisions which split it finally, once again, into two individual cells that contain only one full copy of the genetic information, but is now different from what we started with. And that cycle goes on and on and on, and that appears to facilitate the evolution of all the organisms that do this, because it's got many new possibilities at every generation of responding to the environment. And that may be the key to why the evolution of form in eukaryotes has been much more explosive and much more, and has created much more diversity to our eye, at least, than has happened in prokaryotes. And very importantly, because I'm going to, it was said at the beginning that these talks are about things that are close to our heart, and what I study are the organelles of eukaryotic cells, and one particular class of organelles called the mitochondria. The mitochondria are the organelles, the pieces of cells of all eukaryotes, which carry out fundamental reactions, biochemical reactions, to produce energy from food molecules. I'll describe that in a bit more detail in the next segment. And all eukaryotes pretty much either have mitochondria or they have some genetic or genomic relics of mitochondria that function a bit differently, but that are believed to have a common origin with mitochondria. So you can just look at them as evolved mitochondria. And one particular subset of eukaryotes has got a second organelle like mitochondria, but these are chloroplasts, which empower the organisms that have them, which are plants and algae, to use light from the sun to energize all their biological processes. Chloroplasts and mitochondria are now, or not only believed, but pretty much known to have originated during evolution at the birth of eukaryotes by eukaryotes engulfing, absorbing, processing, and using bacterial species that did the particular things that mitochondria now do for us and all other eukaryotes, and chloroplasts do for plants. So before I break, I'm just going to say a few more words about mitochondria and chloroplasts. Mitochondria are bounded inside the cell by a double membrane. There are about 100 to many thousands of them per cell, and they actually interact, fuse, and divide almost like bacterial cells inside our own cells, having a life of their own. Very importantly, they contain their own DNA. So this is one of the unique features about eukaryotes. They've got not only what you would consider the host cell genome, but they've also got a separate genome inside the mitochondria that encodes some of the functions required by the mitochondrion to do its chemical job in the cell. But there aren't very many of those genes, only about 150 of them in the eukaryotes that contain the most information in mitochondria. Mitochondria everywhere, apart from these weird eukaryotes that appear to have lost their main function, carry out the major energy-yielding reactions of metabolism using oxygen as a final electron acceptor. That simply means they are burning sugars in a controlled way to derive the energy, in the same way that you would burn fuel in your car using the oxygen from the atmosphere as the oxidant that then releases the energy from the fuel to do useful work. And that's what mitochondria are doing biochemically. And chloroplasts are doing something similar. They're found in all plants and algae. And I want to end this segment by just saying what we believe to have happened physically to create this unique constitution of eukaryotic cells. So initially, the eukaryote ancestor, which may or may not have had an endomembrane system, may or may not have had endocytosis, may or may not have had a nucleus, but I've just depicted it here on this slide in the simplest way as if it did have those features, initially came into close contact with a free-living bacterial cell, which was the ancestor of mitochondria. That cell was then engulfed by some mechanism that may have been very similar to what we recognize today as endocytosis. Indeed, there are cells in our body that take up bacteria in pretty much exactly this way. The bacteria are then resident inside the cell, and that's why we call this endosymbiosis. Two organisms living together in harmony, or ultimately in harmony, but one inside the other, endosymbiosis. The bacteria residing inside the eukaryotic host cell are supplied with nutrients. So it's a very good deal from the point of view of the bacteria. Instead of having to move around the environment, very dilute ocean, looking for food molecules to eat, they now have a source of fuel, a ready-made source of fuel, right from inside their host environment, the eukaryotic cell host. I'll come to what those fuels may be in a moment. On the other hand, the host cell developed the means to make use of these intruders. These intruders were eating substrates that the host couldn't efficiently process itself, but the host managed to extract the energy from the bacterial cell in the form of the universal currency of energy that we call ATP. You don't need to worry about the chemistry. ATP is simply a currency. And so this was the essential reciprocity of the deal between the host cell and the ancestor of mitochondria. The host cell, like baker's yeast today, which can actually exist without mitochondria, or certainly without the DNA from mitochondria, they're able to do basic biochemical reactions to break down sugars and get energy, but they're not able to fully oxidize the intermediate product. They just excrete it in the form of ethanol, and we make use of that when we use them to ferment beer and wine and so on. But what the mitochondria were able to do is use that intermediate, pyruvate, completely oxidize it, so a complete combustion to carbon dioxide and water, releasing much more of the energy locked up in that intermediate chemical to make ATP the currency for energy to do useful work in the cell. And then what the host did was simply steal some of that ATP to do things it wanted to do so that both partners benefited. You can look upon that ATP transfer as a little bit like what happens to your bank account every month, that the state of Finland, through the tax office, extracts some of the currency from your bank account to do all the things that are needed to energize the state of Finland, and none of you complain because at the same time the state is providing you and providing all of us with services and materials that we all benefit from. Exactly the same way that the ancestor of mitochondria benefited from the substrates that were partially broken down sugars that the host cell was able to provide. And we can go one better than that. We can actually say what type of cell the bacterial ancestor of mitochondria was, because we can look at its genetic information and compare it with the genetic information of all groups of bacteria, and we find a particular group of bacteria called the Rickettsia, which are very close relatives, or the closest relatives of mitochondria. And very interestingly, even today, the Rickettsia have an endosymbiotic lifestyle, and they actually have their own mechanism for stealing ATP from the host, but they have a completely different protein to do it. So that clearly has evolved subsequent to the initial interaction of the bacteria with the host, and can be done in several different ways. And similarly, we know that chloroplasts with their own DNA, which are found in plants, are related to photosynthetic bacteria called cyanobacteria, and they cluster very tightly in the tree of life with the cyanobacteria. I'll now take some more questions before I go on to the next segment on eukaryote diversity. Yeah.
Stomatolites found off coast Australia, those
Yes.
ancient...
So these are fossil.
Yeah, okay. Are they bacteria only?
You mean, are they bacteria rather than archaea?
Yes, yes.
I have no idea.
Okay.
I think they have been put forward, the existence of those kinds of fossil bacterial communities have been put forward a long time ago, actually, as an argument for saying that organisms do better when they get together. And that was sort of a kind of not more than a metaphor, it was a model for how eukaryotes could have come into existence.
Yeah. Thank you.
Has the sun the optimal set of wavelengths for chloroplasts?
Another question I can't answer. One of the things I'm going to mention in a later segment of my talk is that photosynthesis, in terms of very crude energy efficiency, is not very efficient. There's so much sunlight falling on the Earth that photosynthetic organisms can get by just by harvesting a small amount of it. I guess what you're asking is whether in the course of the evolution of the sun, or indeed of other stars, whether we could find evidence that photosynthesis arose somewhere else or under different conditions. It's an interesting idea. At least it's the sort of concept that one might use to explore that question.
Is there some reason why at each stage these organisms seem to cluster into some sort of individuals? So first cells form, then cells form larger organisms, but things like ant nests where they are almost like an organism, but less, and they maybe can mix with each other. Why is this rare? And can you imagine?
I've just portrayed them in this individualized way for simplicity. I think, as I just referred to, and as is implicit in the origin of eukaryotes, communities of organisms are very common, and I think we can interpret a lot of the evolutionary exchanges that led to eukaryotes as being due to the fact that organisms were indeed living together. And perhaps one of the reasons why archaea and bacteria have exchanged so little genetic information in the course of evolution is simply that they have, at an early stage, adapted to live in really quite different environments. Okay. I want to now focus a bit more specifically on what's happened to eukaryotes since their origin, since they arose. And it's quite clear, as I've already implied, that multicellularity and sex have allowed the evolution of extremely diverse forms, which nevertheless are still eukaryotes in terms of the kinds of cells that they have and the kinds of chemistries that those cells perform, which are, of course, related to all the other organisms as well, but are eukaryote-like in their specifics. So I'd like to go on a little bit further with endosymbiosis. So at the beginning, when bacteria first interacted with and were engulfed by an ancient eukaryote ancestor, they contained their own DNA, their own genomes, coding for all the functions of those cells. Interestingly, rickettsia, which I've already mentioned as an example of an endosymbiont alive today, it's actually the causative agent of a human disease. It causes typhus, which is an epidemic spread through various parasitic roots. And even in rickettsia, the rickettsia doesn't have a full set of genes to live independently. It needs to live within the host either of an insect or of a mammal to survive, because it needs to derive some food molecules that are not found in the outside world at all, are only found inside cells, and therefore it needs to live there. And it's lost the genes that would enable it to live or make to live on or make those other substances. And I already made mention of the fact that mitochondria, whilst they contain their own DNA, they don't contain very much DNA, and that DNA doesn't do very much. It doesn't code for many functions. It's a very restricted or reduced set of genes. So how do we get from there to here. An endosymbiont that had a pretty much complete set of genes to one that's got hardly any. So I think the first step in that was, as has happened in rickettsia, for the endosymbiont to lose its survival capacity outside of the host, and then pretty much its genetic autonomy, because the next thing that is likely to happen is that the precursors to build proteins and DNA and RNA, which are there in abundance in the host, can also be taken up by the symbiont and used to make its own materials. So it's not just burning fuel that the host provides, it's also taking up some of the host's own raw materials to build new things. And that surrender of genetic autonomy is then the beginning of a very long process that appears to have three components. I'm not going to discuss the details here. There are interesting examples of all three of these processes, and they appear to have occurred independently in some cases in the different lineages of eukaryotes, because the tree of eukaryotes also diverged from the ancient eukaryote to form the groups of organisms that we recognize today, and some of these processes happened individually in those lineages. So the first, in the middle, is that the symbiont simply lost its own genes. The DNA for those genes found its way somehow from the organelle, from the bacterial cell or its descendant, into the nucleus. And we know that the cell nucleus has got, or the eukaryotic genome in general, has got this rather interesting property of being able to take up and absorb and integrate bits of foreign DNA quite easily. One of the reasons for that is, in fact, because its genome is so chaotic. So if we just go back to my little model before. Okay, if you just threw a few extra bits of paper into that mess, they would likely be just as well integrated into the whole as all the other bits of paper there. And that's, in a crude way, a kind of analogy for what's going on in the eukaryote nucleus, absorbing genes from the ancestor of mitochondria or the ancestor of chloroplasts. Of course, once they get to the nucleus, those genes have to acquire some new properties, because when they are read and used by the host to make new proteins, those proteins have got to go back to mitochondria. They can't just float around somewhere else in the cell. But it appears that the signature by which a protein can find a new target in the cell is not that complicated. And therefore, even bits of random DNA, such as might be sitting next to that bacterial gene that integrated to the host DNA a billion or more years ago, are quite likely to have been incorporated into it so that they could direct the product back to mitochondria. At least that's what we think, because we find little bits of foreign DNA that do that sitting in front of all of the hundreds of genes that appeared to have followed this route from the symbiont back into the host nucleus. There are also some examples where a function was actually duplicated. The host had the function in its own genome already, and the bacterial invader had its own copy of that, an enzyme to replicate the DNA, for example, or read the DNA information, and many other processes that were found in both types of cell. And here, provided it acquires this ability to go naturally to the new target in the cell, the original gene in the host nucleus could perform the function. So then the bacterial invader might have just lost the gene completely, and it was substituted by the resident gene from the host. And then there are also some examples which I think are very interesting and also give rise to much debate about how much mosaicism has actually occurred and where and when that mosaicism crept in, but we find examples of genes from other bacteria and other archaea that have come into the eukaryotic nucleus and appear to be substituting for functions that were once provided by the bacterial genome. So all three processes have occurred. Nowadays, the organelle genomes that we have, although they're all reduced in scale, are quite different. The chloroplast DNAs are the ones that are, in a sense, the best behaved. They're all pretty much all circular. They're all about 150,000 letters of DNA code, 150 kilobases, and they contain about 100 genes, most of which are either required to make the photosynthetic apparatus, they are the main components of the photosynthetic apparatus, or they are part of the machinery for copying the DNA and turning it into proteins by making ribosomes. And that's another feature, although most of the components that are needed to do that are coming from information in the nucleus, the chloroplast DNA still has some of that information for itself. Mitochondrial DNA is more diverse. In some organisms, it's very small, just a few thousand letters of DNA information. In some organisms, even simple organisms like yeast, it can be apparently large, 80,000 such base pairs, 80,000 letters of DNA code. We're intermediate, we've got about 16,000, and about 37 genes are found in our own mitochondrial DNA. Those genes are unique to the mitochondrial DNA. They don't have counterparts in the nuclear DNA. Without them, we would not be alive, and in particular, our mitochondria could not function. So those 37 genes are essential for life, but they're not the whole set of genes that the mitochondria need to do their chemistry. And plants have got much more exotic mitochondrial DNAs going into hundreds or thousands of kilobases, therefore millions of letters of DNA code, but surprisingly, most of that information appears to be uninterpretable. I won't call it rubbish because we don't really know what, if anything, it does, but the meaningful information, just like in the nucleus, is embedded within a sea of other information that appears to be non-meaningful. And then Reclimonas I've put in there as a particular example of a mitochondrial genome that carries a lot of genetic information in its 70,000 nucleotide letters in DNA. It's got about 100 different genes, so it's a bit like chloroplast DNA and does some of the same things. So I'm going to now going towards my own some of my own interests. I just want to focus on what the human mitochondrial genome does. So this is a cartoon. I don't want to go into any chemical detail, but these green and orange blobs represent the molecular machines that carry out the chemistry of mitochondria, and they are actually representations of proteins that associate together to do that chemistry. And you'll see that we've got a few orange blobs in a sea of green, and that is because the mitochondrial DNA codes for just this small subset of components of these molecular machines, and most of them are depending on genes that are living in the nucleus. So that the whole process of building these machines in a living cell has to take account of the fact that the components are coming from two different sides of a membrane assembly, which is actually two membranes, and they have to cross those membranes and then embed into the inner membrane in a very controlled process, which requires a dedicated machinery. So the whole process is actually much more complicated than it would be in a bacterial cell or in the eukaryotic ancestor. So it's a kind of an example of genetic complexity that has arisen as a result of the eukaryotic condition. And then another complication that we see is something called secondary endosymbiosis. So there are some examples of that, and I'm going to mention two in a moment, where one eukaryote has gobbled up another using a kind of endocytosis, and then transferred most of the genes to its own nucleus or dispense with them altogether, leaving a residue behind of an organelle that is somehow really an organelle from somewhere else. And we know one very nice example of that is the malaria parasite, which seems to be a single-celled eukaryote. It is a single-celled eukaryote, but when its DNA was analyzed, starting from the 1980s, it became clear that this parasitic single-celled organism had a relic of a chloroplast genome. And so that precipitated a lot of debate. Is this parasite actually a plant? Well, it's not a plant in terms of its physiology, but it contains a genetic relic which appears to have been acquired in a secondary endosymbiosis from a photosynthetic organism, and then all the genes for photosynthesis have got thrown away, and it's just it just contains a small packet of genetic information which now encodes something different than a chloroplast. It's actually another organelle that is involved in the ability of the parasite to invade other cells. Here's an example of an much earlier stage in such an evolutionary process. It's an animal, a sea slug, that is green, and it actually uses, the green is due to chloroplast. So it's an animal that has chloroplasts inside its own cells. How can that be? Well, the answer is that the chloroplasts are not in any way encoded in the genome of the sea slug that is green, but they've actually been absorbed into its cells by endocytosis after the sea slug has eaten photosynthetic algae, destroyed the rest of the alga as its food, and incorporated the chloroplasts into its own cells. There was a lot of debate early on as to how these chloroplasts can survive. The answer is they don't survive indefinitely, and they're not passed on to the next generation. They're taken up anew in every generation. But there are some host proteins that appear to facilitate in some way the survival and operation of these photosynthetic objects, which are providing energy for the animal. So you can consider this as a very early stage of a secondary endosymbiosis. Despite these complications, the eukaryotic tree is quite well organized. Apart from that bunch of single-celled organisms where there have been quite a lot of secondary endosymbiosis by one eukaryote or another, or other archaebacteria or archaea and bacteria, then the other groups of eukaryotes are pretty much uniform and not communicating with others genetically like the sea slug is. So the fungi are fungi just like bacteria are bacteria, or fords are fords. The animals are animals, the plants are plants, and so on. Okay. Oh. Okay. I'm going to go quite quickly through the next section. One small point I want to make is that evolution, although evolution is an ongoing process, so we are, in our own eyes, but not the eyes of a sea slug, we are the pinnacle of evolution. But evolution is going on in all lineages of organisms all the time, and doesn't only go in the direction of increasing complexity. Here's an example where it went in the opposite direction of simplification. So the baker's yeast, which I've already referred to, that we use to make beer and bread and so on, is a simple single-celled organism that's actually lost many of its genes during the course of its evolution, but it's actually a close relative of these much more sophisticated and complex-looking organisms, such as the morel, the scarlet cap, and other fungi that are living in our forests. And we see the same trend in plants and in animals that become parasites, surrender much of their autonomy, and just depend on other organisms. So their genomes get less complex, and their biology gets less complex, or more, in another sense, more specific, but less complicated. But this is not reverse evolution. This is evolution. Evolution can go in the direction of increasing complexity or decreasing complexity. The only thing we can say is that the genes that were lost will not be recovered again other than by some kind of symbiotic, endosymbiotic event in the future. The animals and plants that we see around us, as I said, are all eukaryotes made of eukaryotic cells, but they're multicellular. The combination of multicellularity and sex has given rise to this huge diversity of forms. So even closely related animals can have very different morphologies, very different lifestyles. And that includes the organisms that we're perhaps most familiar with, mammals and birds. I'll take questions now before I go on in my last segments to talk about disease. Yeah.
Do mitochondria replicate just by copying, or do you have some gene exchange? And does one human being have similar mitochondrial DNA all over the place, or?
Yes, mitochondria are, and their DNA are vertically inherited through the egg. So I'll mention this complication in more detail in a moment, but essentially all of the mitochondrial DNA in your body is unique to you and is different from in small details from the mitochondrial DNA of the person sitting next to you in the audience, unless they happen to be your identical twin, which they don't seem to be.
Can I pose a question here? Because I have an identical twin and she's not me, so as a scholar of humanity, I of course wonder, so how much of what I am is the bacteria and the evolution and the mitochondria? Maybe when you think about yourself, how much of yourself is this and how much is...
That's a philosophical question, because, as I've said, the mitochondrial DNA is a tiny packet of information compared. So the mitochondrial DNA has got 37 genes. The nuclear DNA has got upwards of 20,000 genes. And it's much bigger in terms of its total length of DNA code. The human nuclear genome is 3 billion letters of DNA code. The mitochondrial DNA is just 16,000. On the other hand, if you take the DNA from a human oocyte, because the human oocyte is a very big cell, and it's only got one copy of the nuclear DNA, but it's got lots of mitochondria, you'll find that almost half of the total DNA in the egg is actually mitochondrial DNA by mass. So it's a question, I think, of philosophy, whether you regard the quantity in your egg that gave rise to you versus the total DNA that you now have, or the informational complexity as being more important. But you're not exactly the same as your identical twin, as you know, and of course there are many reasons for that. One of the main reasons is that the DNA in the nuclear genome, at least, is responsive to the environment, undergoes some particular modifications which are responding to the environment, and therefore set itself in a slightly different way in each individual. Yeah.
You are using the word chaos differently than mathematicians use. Is there no words to replace for you?
Okay. I've used it just as a word to mean superficially disorganized. As I've said, there is an organization within that overall appearance or veneer of disorganization, but to somebody who was analyzed, to a person who was schooled in literature, the bacterial genome appears to be a beautiful poem, whereas the DNA of the human DNA is just a series of random words which are then embedded within long chunks of text that are random letters. And so it's very difficult to make sense of unless you know the rules. But I agree, chaos, I'm just using it in the everyday sense, not the mathematical sense. So I'm going to have a brief segment now where I'll talk about heat and light to a lesser extent. I already mentioned that we have this concept of energetic efficiency in which photosynthesis, because most of the light falling on a leaf or falling on an algal cell is not actually getting turned into chemical energy to make ATP, but it's just getting lost. The energetic efficiency of photosynthesis, at least at the cellular level, is not very high. The mitochondrial fuel-burning engine is actually quite efficient compared, for example, with the internal combustion engine. About half of the chemical energy of its fuel is turned into ATP to drive useful work in the body in mitochondria. Whereas the internal combustion engine at most does about 35% efficiency, and all the excess energy is then dissipated as heat. Interestingly, electrical engines, which are used now in the next generation of cars, are much more efficient than either of those. But there are some cases where the heat production, which is just really it's a byproduct of the chemistry, and it's a necessity really because of the way the chemistry operates, and that it's this most efficient way of burning fuel has evolved. But nevertheless, there are some specific biological contexts where the release of heat by mitochondria is actually of great physiological importance. I'm going to mention just two of them. One is in our own bodies, and this has also only been recognized properly in the last 25 years or so, that there is a class of fat cells in our body. It may be a little bit incorrect even to talk about them as fat cells, because most of the fat in our body, the abdominal fat and so on, that most of us would be happy to get rid of if we could, is composed of so-called white fat cells. White fat cells are really just storage cells containing fat to burn on a rainy day when we can't get carbohydrates and other food. There's this other class of cells called brown fat cells, which were overlooked for a long time because they were thought to be only in hibernating rodents or in newborns, and that there isn't very much brown fat in an adult human. But that's because it is so efficient at doing what it does, which is to produce heat for the body when it's needed, that it could almost pass unnoticed. In most humans, brown fat is just, as in the slide, is just in the back of the shoulders and in the neck. What brown fat mitochondria do when they're activated is to produce heat, not ATP. So they, instead of, like the standard mitochondrial metabolism, instead of producing ATP to drive nerve transmission, to drive the growth of the cells, to drive muscle contraction or the beating of your heart and so on, the mitochondria of brown fat is dedicated to produce only heat. And it does so by short-circuiting the whole system of energy metabolism that I don't have time to go to in detail, which involves, in a normal mitochondria, a kind of a charge separation. It's making a kind of electrical, very crude electrical cell inside or across the mitochondrial membrane, which has a gradient of protons, which create an electrochemical gradient, which then drives the synthesis of ATP in one of the components of the system. But instead of the protons being used in brown fat mitochondria to make ATP, they are then simply short-circuited back into the mitochondria in a futile cycle, with the energy from their pumping out being then dissipated entirely as heat. That protein is called UCP1. That stands for the uncoupler protein, because it uncouples fuel burning from useful work. And here's another example in plants. We mostly think of plants as cold or cold-blooded kind of organisms that are just at environmental temperature. But many plants are actually warmer than their environment. And in the introductory slide of this segment, I showed another example where the skunk cabbage is poking through the snow because it's also releasing heat. All organisms release heat if they're doing metabolism, and that means that some of the energy is always getting wasted as heat. But what this particular group of plants does is to actually use a similar system to the uncoupling protein. Its chemistry is a little bit different, but the effect is the same, to heat up the female parts of the flower so that they will then volatilize various nasty compounds, nasty smelling compounds that are delicious to flies, which are then attracted to the flower, crawl onto the flower, and shake around the pollen, or bring pollen from another flower, which can then be deposited to fertilize the egg cells, the germ cells in the female part of the flower. So this is it's a physiological system connected with sex in the flower, but it requires the specific production of heat in mitochondria. And that's accomplished, in this case, by a slightly different system. Instead of dissipating the proton gradient created across the membrane, in this case, the proton gradient isn't even created, or it's actually in a much reduced form. Fewer protons are being pumped out per cycle of oxidation of fuel, because the plant introduces another protein into the mitochondrial membrane that does the same redox chemistry, but doesn't do the proton pumping, which leads to the production of ATP. So again, it wastes the energy as heat, and that heat is then used to do what I just described in the sexual cycle of the flower. Here's something that we stumbled into in my own lab and that of a collaborator recently. It's an unpublished paper, which is actually out to review at the moment, but I felt I just wanted to mention it. We decided to measure the temperature of mitochondria. We already know that mitochondria are emitting heat, but I think the traditional view was that the cell is such an efficient heat-conducting system. It isn't, but we thought it was, that the heat produced by mitochondria would just be dissipated in the cell, and the whole cell would be at a constant temperature. But actually that's not true. The mitochondria turn out based upon heat-sensitive fluorescent probes, chemical probes that we've introduced into mitochondria and followed their fluorescence under different physiological conditions. It's clear that the temperature of mitochondria in our cells, even cells that are bathed in a medium at 37 or 38 degrees, the mitochondria are 10 degrees hotter. So they're much more like little radiant fires in our cells than like the efficient heat distribution system in Roman baths. The whole issue of temperature in biology is the subject of much debate. Most organisms, including certainly all those that, well, not all of those, almost all of those that live in the oceans, are so-called cold-blooded or poikilothermic organisms which exist at the temperature of their environment. So if you go fishing in an icy lake in Finland, the water is at 4 degrees underneath the ice, and the fish that you're trying to catch are also pretty much at 4 degrees. And it's always been thought that this enables the organisms that do this to avoid wasting energy to adjust to an arbitrary temperature that's different from the environment. Whereas birds and their dinosaur ancestors and mammals have evolved a system whereby they maintain their bodies through mitochondrial heat production at about 38, 39, 40 degrees. Most of those organisms live in the air or on land, where heat conduction from the organism is much less. So that's one reason why this probably could evolve. But what's special about 40 degrees is very unclear. If that's just simply some kind of balancing temperature between heat production from mitochondria and loss to the environment, you'd expect that it would vary in organisms that were of different sizes or living in different environments, in the tropics or in the polar regions. But emperor penguins that I illustrated before are also living, their bodies are also at this temperature. So even though the outside environment can be minus 70 degrees Celsius. So what's special about 40 degrees? I don't have an answer to that, and I'd be very interested to hear if anybody in the audience does. That's the end of this short segment.
When the brown fat cells are activated, do they help to burn also other fat in the body?
Ultimately, yes. So if you're in the cold, where the brown fat is activated and is very important, if the brown fat cells will run out of their own fuel, they'll take in fats that are secreted from other cells, including the white adipose cells, or new fats that are produced from carbohydrates in the liver. But if you're starving and in the Antarctic, then your brown fat will be burning pre-existing fuel. That's what happened to the early Antarctic explorers, which by and large died of starvation. Okay, let's go on. I'm going to take a very quick cook's tour around another pet subject of mine, which is the main topic on which my lab works, which is mitochondria and disease. So the eukaryotic constitution, as I've described it, with mitochondria as a separate organelle and mitochondrial DNA as a separate packet of information, creates, although it's a very good arrangement in terms of evolution and fueling efficiently the organisms that have that, including ourselves, it also creates vulnerabilities. Mitochondrial DNA is essential for life, so we can't live without it, so that's our first vulnerability. And also, quite interestingly, perhaps a relic of the early phase of endosymbiosis, even today, the mitochondria in our cells still retain the ability to signal suicide to the rest of the cell. That seems to be surprising, but you might think that would be a very good strategy for an endosymbiont that got trapped by an unfriendly host. The best way to deal with that situation is to kill the host and get out and go somewhere else. And this whole process of programmed cell death, which the mitochondria appear to instruct, and which does appear to be, at least in the form that we understand it, a uniquely eukaryotic invention, this process seems to be, or mitochondria seem to be central in this process, in deciding the fate of cells. There are all kinds of reasons for which cells might decide that it's better for them to commit suicide than to continue, but the mitochondria have a crucial role in making that decision. And if the mitochondria are not working properly, or if the cell is not delivering to the mitochondria what they need, the mitochondria will activate the cell death program. I've already mentioned the complications arising from having 1,000 copies or more of mitochondrial DNA and only 1 or 2 copies of the nuclear DNA. Every gene for one of those green blobs in my cartoon has to, in a sense, work 500 times more actively than a typical mitochondrial gene, because the mitochondrial DNA is dispersed over the whole set of mitochondria, and the nuclear gene has to feed all of them. A complex assembly process, as I mentioned, has to be invoked to build those molecular machines that the mitochondria use to do energy metabolism. And mitochondrial DNA now depends entirely on nuclear genes for its own maintenance and faithful replication. If that machinery doesn't work, then the mitochondria are compromised, and then the cell is compromised. And the absence of non-coding, so the mitochondrial DNA of humans, as I said, is very reduced in size. And so there's almost no non-coding information left. Even more than the typical bacterial genome, the mitochondrial genome of humans has got rid of all extraneous information. It's like an ultra-refined modern office compared with the nuclear genome. But that also creates a vulnerability, because any mutation in any letter of the DNA code of mitochondria is likely to be damaging, whereas most of the letters in the nuclear genetic code are immune to those effects. Also, and I think probably the most important aspect here, is that the mitochondrion itself, because it's doing all these energetic reactions, it's interacting a lot, or its molecular machines are working with oxygen, which is potentially very toxic, creating free radicals from oxygen, which can damage proteins, can damage membranes, and can damage DNA itself. And that's all taking place in the kind of hothouse, literally, of mitochondria, which is also hotter than the rest of the cell. So all these potentially damaging chemistries are taking place in the very environment where the mitochondrial DNA is located. And that makes it susceptible to damage. Maternal inheritance is something that is unique to organelle DNA. It occurs in virtually all animals, and certainly in humans. As I already said, the human egg is full of mitochondria and full of mitochondrial DNA, and only the mother's DNA is passed on to the next generation. the sperm mitochondrial DNA is actually actively destroyed after fertilization. So this unique maternal inheritance means that mitochondrial DNA is not experiencing the evolutionary virtues of sex, which all the other genes in the human genome are able to take advantage of. And that again means that it's less adaptable in certain ways. And so mitochondrial disease, because the mitochondrial DNA is so important and the mitochondria are so important, takes many different forms. Primary mitochondriopathies, where the mitochondrial DNA itself is damaged, or where some of the genes in the nucleus that are supposed to process mitochondrial DNA or contribute to the molecular machines of mitochondria, these can affect a huge variety of different cell types in the body, and as I put here, pretty much any cell in the body, and often many all at the same time. So mitochondrial disease is very diverse. It's quite rare, so individual types of mitochondrial disease arising from specific genetic defects are individually rare, but collectively they're quite significant. They are pretty much as common collectively as other common inherited diseases. But they can affect not only the tissues that we know that are very dependent on energy, like the brain, the heart, the eyes, skeletal muscle, but also the kidney, the pancreas, the liver, and pretty much any organ in the body. And then something that's only become recently properly recognized, because mitochondrial function is essential to the life of the cell, mitochondrial dysfunction, which may be secondary to other mutations in other physiological systems, or to toxic insults from the environment, such as ingesting smoke through tobacco or malfunctioning vehicles in Mexico City and Beijing that are polluting the atmosphere, those are also then crucial components in disease processes. So we now recognize that diseases, common diseases like Parkinson's disease, diabetes, stroke, many forms of heart disease, probably Alzheimer's, dementia, hearing loss associated with age, and lung diseases like COPD have a major dimension of mitochondrial dysfunction, which also gives us a potential avenue in therapy, because if we can correct the mitochondrial dysfunction, even if it's not the primary cause of the disease, we can alleviate the symptoms and arrest the disease process. A cell without a mitochondrion is like a car without an engine, and it's not just lacking energy, because other bits of the car, if they're not working properly, can be a drag on the engine and lead to the production of toxic byproducts. So in mitochondria, this cartoon is just a summary of what they are. Mitochondria are at the heart of pretty much all metabolism for breaking down food molecules. All those pathways are interrupted when mitochondrial oxidation isn't working. We have production of oxygen radicals when the whole system isn't working properly, and electrons are passed inappropriately to oxygen to create these damaging moieties called reactive oxygen species. If the oxidation reactions are not working properly, the body has to go back to an earlier, evolutionarily earlier chemistry, a bit like the production of ethanol in yeast, but in the human body, we don't produce our own ethanol, we produce lactic acid, which then leads to the acidification of the tissues, which then does damage of its own. And all these defects can lead to the inappropriate induction of signals for cell death. So I just want to mention very briefly an approach that my lab is taking to design a strategy, if not to recover the energy production, but to alleviate all of these secondary defects of mitochondrial dysfunction, which can occur for a variety of reasons, genetic, toxicological, and other kinds of damage. And that is to take that very gene that I already introduced in the case of the flowers that produce heat, the alternative oxidase, which we can use to bypass a malfunctioning part of the mitochondrial energy system. Although this produces heat instead of ATP, it actually liberates the rest of the system to produce ATP and can alleviate some of those other secondary consequences. I'm going to skip over the rest of that. I'll answer some specific questions on that if you want to ask some. And at the end of my talk, in the next couple of minutes, I'm going to focus on the future. Any questions about mitochondria and disease?
So I've heard that in some cancer, the mitochondria undergo a change after which they cannot use other than carbs as energy source, and so this cancer can be killed with a ketogenic diet.
I've avoided cancer completely from my list of mitochondrial diseases because whilst what you've just said is perfectly true in some cancers, there are other cancers where the exact opposite is true. So it appears that mitochondria can be indispensable for a cell to become a cancer cell and can also prevent the production of cancer in specific contexts. And like many pathways that are modified or abrogated in cancer, we don't fully understand how those two systems or how those two outcomes are brought about in general terms. So I think trying to interfere with that, just like trying to interfere with many malfunctioning pathways in cancer, is fraught with difficulties.
I'm wondering about the lactose intolerance because we are milk drinking people and more people are having that issue.
I think, well, this has nothing to do with mitochondria because the enzymes to degrade lactose are actually not in the mitochondria anyway. And lactose is different from lactate, which is an intermediate product actually made by the body itself, breaking down sugars. And I think lactose intolerance is increasingly recognized, partly because we're just drinking more milk products. I want to say some general words about the future of evolution. This seminar or symposium is about becoming, so I've talked a lot about how we got to be like we are and how all the other different types of cells around us are like they are. But we now have reached a point in cultural development where we are able, in principle, to alter the course of our own future evolution. So I just want to pose an ethical question to you. If we're able to do that, let's say, for argument's sake, I haven't had time to talk about the alternative oxidase in much detail. Let's say we were able to introduce the alternative oxidase from another organism because we don't have it, so that it functions in our mitochondria and protects us against many of the mitochondrial diseases that I've just mentioned. Alzheimer's, perhaps, Parkinson's disease, heart disease. Even more starkly, if we would take one of the genes that we know protects mammals against cancer, which is present in multiple copies in some long-lived mammals, and add extra copies of it to the human genome, so as to reduce the risk of cells becoming tumors by loss of this gene if there are more copies of it to rely on. Would it be ethical for us to do that? What is more unethical? Is it unethical to play God, to interfere with evolution, egocentrically promoting our own, in the future, perhaps immortality, contravening what most of the Western religions would regard as an absolute ban on seeking to manipulate human life? In the biblical story, the angels with flaming swords were placed all around the tree of life to prevent us gaining that knowledge and using that knowledge. Is it unethical for us to do so? Or is it unethical for us to deny treatments that would alleviate or prevent human suffering? Directing evolution for our own purposes is fraught with troubling consequences, and there are many works of literature which have highlighted some of these dangers and how our own intellect can lead us astray into damaging as well as beneficial consequences. And indeed, even in so-called democracies, we can ask who we might trust to make decisions to mobilize genetic technologies for global good. I think the answer has to be that we need to learn about how evolution works, we need to learn about biology, and we need to then inform our policy-making bodies of the facts as we see them to make those decisions. And there's a very nice example of this having recently happened in the UK. It will undoubtedly happen now in an accelerated form in other countries, where mitochondrial DNA has actually become the first such target. The process known as nuclear transfer, in which genetic information is moved between oocytes, it's called, in a sense, I think it's misleading, but it's called the three-parent embryo, has been put forward and now approved in the UK as a technique to prevent the development of mitochondrial disease for mothers that contain only mutated mitochondrial DNA that would give rise to children having clear-cut lifetime defects in their energy metabolism. But of course we could ask whether if we let this process go on without proper regulation, we're simply going to end up bequeathing the entire world to bacteria and archaea, and there won't be anything left of us and perhaps of other eukaryotes either. So I'll end by just acknowledging the many sponsors of my own research and by saying just a brief word of tribute to those in Turku who have recently suffered at the hands of people who want to deny us this freedom to inquire and to promote the truth. Thank you.
You've been using lots of images and you've been using lots of metaphors, especially from the car industry. But what is there a reason to consider arts and science seriously?
You mean the interface of
Yeah.
arts?
In your work.
I brought together, to some extent, biology and engineering. I think I would address that question to the arts community, where interpretation is much more fluid, but where you can certainly, in the medium of the arts, you can express ideas that are actually quite difficult to express in words, whereas we, natural scientists, use words and concepts built from words all the time. As you've said, I've used metaphors.
It's of course quite alarming, the last images, and the idea that we would actually might reverse the situation and become bacteria again, and especially I think the question of who gets to take these decisions on what should be manipulated and what not. I mean, how dystopic are you? Do you think it is still a process that we can regulate and guide, or I mean, is there a possibility to make these kind of regulations worldwide on what we can and what we cannot do, or is that time already past?
I don't consider myself to have any greater expertise on this than any other member of the audience. It depends, how I'm feeling every morning. I guess like most of us, we see things in the world around us that are very troubling, and we see things around us in the world that are reasons for hope. Most of the time I'm an optimist. I would say in some ways human history is a little bit like the eukaryotic genome. It's superficially chaotic, but somehow there is some kind of hopeful, organized message that comes out from history in the same way as from the genome. And maybe we, despite not having a very organized framework for making those decisions that will get made in the long term will be for our benefit. But on a bleak day, I can take the opposite view.
I'm not sure whether I can put this question into words, but you touched on viruses being like packages of information moving into cells, and then the cell itself being like a community, and multicellular organisms being communities on the next level, and then we have like ants and bees and we have society. which is like all different biological levels of communities. And then I just started thinking of, let's say, the internet and artificial intelligence, which is also information moving freely outside of bodies. Would you consider the world we live in today like being a biological next step?
Well, I mean, you've used some of the same words. I mean, we refer to computer viruses or to concepts on the on the internet being viral or becoming viral, which means they're rapidly propagating. And indeed, most of the viruses in the communications media are, like most of the biological viruses, actually rather benign. They're just a little bit selfish, or they've been, or they have evolved to serve the selfish ends of some other persons or groups of persons. So they don't actually do much damage to your computer. And indeed, you know, when you hook into the site of a newspaper, they now ask you whether you're happy. Well, you don't really have any alternative to have some of these viruses that they now call cookies, these benign viruses that are scanning your computer for keywords all the time. So yes, I think there is a very close analogy.
So my question is a bit medically orientated, and you mentioned that there are several diseases, for example Alzheimer or Parkinson, where the mitochondrial dysfunction exists.
Yeah.
So what is the stage regarding the drug development? Is this taken further? Can we find anything to make mitochondrial dysfunction better in Alzheimer?
Drug development, there are two kinds of drug development, and I think there's a lot of debate in pharmaceutical industry as to which of the two is the more fruitful approach. One is to identify clear biological targets, molecules, pathways, pathological variants of those, which can then be targeted by a well-designed chemical entity. And then there's the other approach, which seeks to screen all chemical entities at random, but just with a biological readout for improved health of some form. And so, first of all, I don't know which of those two is, in the end, going to be more fruitful. I would guess the latter, but I'm not sure. So, if it is the latter, then drugs targeting mitochondrial dysfunction should aim at alleviating mitochondrial dysfunction or at alleviating Alzheimer's. In the end, it doesn't really matter how Alzheimer's is caused or how a drug works, if it will work. So I think that because our understanding of some of those diseases, which are very complex, is still quite rudimentary. There are thousands of papers published on Alzheimer's dementia every single year, and they're extremely confusing and contradictory. We have no clear pattern that emerges, just a few generally accepted principles. So to try to build a drug for Alzheimer on the basis of that sea of contradictory literature at the moment is extremely hazardous, I think.
Thank you.
Mitochondrial dysfunction included.
Okay, thank you. Thank you for the audience for being